Hello, welcome to my audio-electronic presentation for Antioch University ES 696 with Professor Sarah Baucus. I'm Christopher George, and I'm going to give you the story of my internship this semester at MIT Sea Grant with Dr. Robert Vincent. The focus of our study in this internship was the river herring, which is a local favorite, of which there's two kinds, the alewife and the blueback. Every spring in a famous migratory spawning run, they run up the rivers from the bay or ocean habitat to a freshwater pond in order to spawn. If you check out this video, you can see some underwater video of them on the move. There they go. So our interest is to understand how the marine ecosystem connects with the terrestrial through the life cycle of this fish. And to give you a bit of the natural history background, in our field site, these fish are moving from Cape Cod Bay in Plymouth, seen here, up a tidally influenced river to the Tidmarsh Farm through Tidmarsh Farm West, which is a massive ecological engineering restoration project of a restored cranberry bog habitat. And they run up through this. If you look here in the middle, there's a, right there, a antenna that we built, and into a freshwater pond where spawning will occur by the thousands. There's the fish. And this photo was taken by a property owner who owns pond front property in the spawning pond, which is Fresh Pond in Plymouth. So the location of our lab work is here at 12 Emily Street in Cambridge. And it's a brand new lab. There it is on moving day, where I did my work with one of the Sea Grant professors. And our study is twofold. One was to prepare samples for stable isotope analysis with the end goal of creating food webs to help understand the ecological connectivity between marine and terrestrial environments through trophic transfer of energy and calories seen here. So basically, we would prepare samples, drying fish, liver, flesh, and mucus samples, as well as other wildlife from the field site. And they would be dried, ground up, run through spectrometric analysis up at the University of New Hampshire. And from that, this chart, as you see, or one like it, can be made to draw more informed conclusions for management of fisheries and natural resource. And the other half was in the field this season constructing PIT tag antenna and doing PIT telemetry. PIT stands for Passive Inductance Telemetry. As you can see here is one of our antenna. And basically, the idea of PIT tagging is to tag a fish with what amounts to pretty much a glass bead encasing coiled magnetic material that will pass through one of these antennae as the fish swims up the stream. And the antenna is simply a coil of wire charged with electricity that can be tuned to sensitivity to the interruption of that field, which can be read by computer and will get the signature of that tag. So you can get location and time and item number from the fish. And this is the same technology used by highway departments for E-ZPass. So we're setting that up to see how in a newly created habitat. Remember, this used to be a cranberry bog, and now it has this man-made freshwater stream that's newly being used by herring. We want to track their movements by telemetry tagging. So the challenge this internship was to get this equipment in the field and the fish tagged. To do so, just a footnote, we started at Silvio Conti USGS Lab out in Western Mass, where we consulted with some professionals and colleagues of Rob's in the field to get a background on how to set up the computer and how to build the antenna. So for the rest of the presentation, allow me to take you through the story of how myself, a classroom graduate student, adapted to this cutting-edge lab at MIT and to field work in what amounts to the largest restored habitat on the East Coast. So I hope to show you a little bit about what I learned in the lab, in the field, and the experience I gained in stakeholder cooperation. Laboratory. I learned the lab can be a time-consuming place, and work can be repetitious, but the excitement that comes from the privilege of discovery, even a small discovery, and to work with something as varied and exciting as a real spectrum of pond fish and herring was, a lot to carry the enthusiasm for the work. Look at those guys. For instance, if you'll see, this is our, these are our samples, which will be dried and processed, and that's a liver, a fillet. And in the bottom right of that little tin is a piece of fish mucus, which is an exciting thing to behold because it is an experimental substance being used for isotope analysis, because it's less invasive and certainly non-lethal to the fish, that in the future, with calibration, perhaps mucus can be used for this kind of study instead of full harvest of the fish, as you see here. Another thing exciting in the laboratory was the gut content analysis, which goes with a whole suite of other data that we were collecting alongside our target of isotope samples. Stuff like size, weight, and stomach contents that just came along with the organism. And contrary to what I found in my literature research, which expects the herring to spawn on an empty stomach, I found some to be gorged with shrimp and a little bivalve, as you see here. And one other thing I learned in terms of laboratory praxis was, you'll notice this photo is under magnification through a microscope, and that's just a handheld camera phone. So smartphones are a great resource, and they're right in your pocket, and all you got to do to take high quality photos is just put the smartphone right up to the camera, to the microscope, and with a little patience you can get an excellent picture. Here's me preparing for pit tagging by getting them all in a row into that piece of styrofoam to be taken into the field. Field skills were numerous, and you can see a little exposition of the environment, and here's Rob and I working on an antenna, working away there. So field skills took us out in the field, cooperation, troubleshooting, working with other volunteers to get a multifaceted and many moving parts of this deployment of 8 different antennae into different locations across streams in the Tidmarsh site, and using these. Here's a close-up of the tuner. So a big portion of this project was getting out in the field. And then we had to catch the fish for tagging. So fishing was exciting. We tried numerous different setups to endeavor to catch the fish at first, including technique of block seining, and then we dropped the net. Here's a weir which was set up, great expectations, but unfortunately overnight it breached. So what we settled on eventually was something like this, and as you can see, we're plucking out fish with a net, setting them up to be tagged, and inserting the tags, as you can see here. And the biggest part, last part, would be stakeholder cooperation that I learned. And as you can see, a lot of people are attracted to this high profile and high level restoration site, and there's great science going on. So I was impressed at how many different collaborators there were. And one thing I observed was when there were professional collaborators, some were Dr. Vincent's personal colleagues that he had worked for or with at one point or another. And not only were there dedicated volunteers putting in hours, but Dr. Vincent's colleagues from around Massachusetts, many of them busy PhDs, were happy to give their time and learn a new technique and spend a day in the field. So I saw a lot of collegial cooperation, which really amazed me. And then lastly, I'd just like to hit on some corollary experience in environmental ed, data entry, and other minutiae. So I've noticed with scientists a great privilege is to be able to share their work with children, young school students. So it's a great justification for the work. And though I've taught youth before, this was the first time I got to do it embedded within a project like this. So that was excellent. I learned a lot about data entry, and it's good practice to have a Write in the Rain notebook or something waterproof to write on, and to use pencils, because when the water gets into that 8x11 computer paper, it's going to get wet, and unless you're using a mechanical pencil, which has the advantage of a standardized lead size, sometimes it can be hard to read your own writing. So I recommend the Papermate, and to go with this good old plastic mechanical pencil. Also, Write in the Rain notebooks can be drilled through either to tether to the site for logging by volunteers, or for security if that's needed. And then in smallest minutiae, furthest from scientific importance, was just quality of experience, which I realize a lot of people are into. There were a lot of students now in graduate school, and even professionals in the field value the environmental sciences for the quality of the work. And here's a Snark boat which we used to get across the streams in Tidmarsh, and it was turned out to be great. We tried a couple things, a canoe, and this was property of Tidmarsh itself, and turned out to be excellent. Just experience with this 1970s model sailboat. So special thanks to Sarah Baucus, Dr. Vincent, and all the volunteers and collaborators at MIT Sea Grant and Tidmarsh, who I learned from and had the privilege of working with this semester. Thank you very much. Take care.